In this video, we are starting the second part of the Pharmaceutical Biochemistry class, concerning the ways that nucleic acids store and transmit information. And specifically in this module, we're going to cover an overview of the structure of DNA and what is called the Central Dogma. As future pharmacists though, your first thought is probably, why should we care? Well, I'll first give you one example that shows how this area impacts patients in the clinic. One of the clinical areas we will come back to again and again in this section is cancer, because this disease is caused by errors in the genetic material within cells. Disease progression is caused by a form of evolution by natural selection, and we also treat cancer with drugs that attack DNA itself, and also by inhibiting DNA replication and by inhibiting nucleotide and RNA synthesis. Therefore, to understand cancer chemotherapy, it helps to understand evolution, the structure of DNA, how DNA is synthesized in the cell, and how nucleotides and RNA are made. I'll show you what I mean. Your body is made of about 100 trillion cells, and every time one of them divides, there is a chance that the genetic material inside that cell could be copied incorrectly, making one of the new cells a bit different. After many times of this happening, some cells will be able to divide faster and more often. Over time, you naturally get more of the cells that can divide faster, and they then make up more of the cells in the local tissue than the unmutated normal cells. If additional mutations allow cells to divide even faster, then those cells will outcompete the previous versions and normal cells. This is a manifestation of evolution by natural selection. Cancer is a group of diseases where mutated cells, like this, have begun to divide uncontrollably, with different cancer types classified by the location in the body and/or the cell type affected. This can be a life-threatening situation because these cells are generally no longer doing their normal function, and their uncontrolled growth can threaten the function of other tissues and systems. So the question then arises, how do we treat cancer? We want to kill cancer cells, but they are still human cells which are very similar to the normal human cells in the body that we don't want to kill. And if it sounds like a difficult task to selectively kill cancer cells, you are right, it is. Many anti-cancer treatments end up killing a lot of normal cells as well as cancer cells, and that is why these treatments often have a lot of bad side effects. How they generally work is that they target things that a lot of cells need to do, but cancer cells do more. For example, we know that cancer cells divide uncontrollably, and one of the processes that needs to happen during division is that a cell's genome stored on a DNA molecule has to be copied. So if we have drugs that mess up this process, then we can also stop division. Drugs can mess up the process of DNA copying a few different ways, including inhibiting the process of DNA replication, the copying process itself, or inhibiting the synthesis of nucleotides, the building blocks of DNA. So you can see that if you understand these processes, then you will also understand how drugs in these categories work. And if you think about how these processes also affect normal cells that need to divide rapidly, it is easy to understand why they cause bad side effects like low white blood cell count and hair loss. So in that example, I mentioned that a cell's genome is basically a bunch of genetic information that is stored on a DNA molecule. But what is DNA? Well, DNA is an acronym standing for deoxyribonucleic acid. It is a sort of technical chemical name, and we'll go more into what it means later. For now, I will say that there is another type of nucleic acid in cells called RNA, which stands for ribonucleic acid. What do these molecules do? Well, they are both polymers of nucleotides in the same way that proteins are polymers of amino acids. They are both special in that the order of different nucleotides in these polymers stores information. In addition, RNA can form complex 3D structures that perform chemical catalysis, just like proteins. But what is the difference between DNA and RNA? We'll go into the detailed chemical differences later, but for now the main difference to understand is one of stability. DNA has a half-life of 521 years, and you can even recover it from mummified mammoths and other extinct animals. RNA, by contrast, breaks down in a matter of minutes. So knowing this, it makes sense that DNA would be the molecule that is used by cells for long-term storage of their genetic information. But then why do we need RNA, and what information is being stored in DNA? Well, you already know from Professor Lee's lectures that proteins do most of the work in the cell, and it turns out that the amino acid sequences of each protein the cell needs is encoded in its DNA. But it also turns out that you can't directly go from DNA to protein. This brings us to what is known as the Central Dogma in biology. Shortly after the structure of DNA was worked out, one of its discoverers, Francis Crick, proposed that there are certain ways that information can flow in a cell. When a cell divides, we copy its DNA to a new molecule, and this is called replication. We can't go directly from DNA to protein, but we instead have to first copy the information from DNA to RNA, and this process is called transcription. The information encoded on RNA can be used to make proteins, and this process is called translation. Now, the use of the word dogma here is strange. The definition of the word is basically something that an authority proclaims that should not be questioned. It is therefore not very scientific, because in science we change our model of the world based on new evidence. Crick admitted after proposing this name that he was not entirely clear on the definition of dogma and that it was inappropriate. However, this is the name that we are stuck with at this point. In fact, the Central Dogma has been updated a few times, as we will see, but this main part has stayed the same. But how is information stored on nucleic acids? Well, let's first look at the primary sequence of a DNA molecule. Just like with a protein sequence, we have different names for the ends so that we know which way round the molecule is. However, instead of an N and a C terminal, we call one end the 5' end and the other the 3' end. By convention, you write a DNA molecule going from the 5' end on the left to the 3' end on the right, in the same way that you would normally write a protein sequence from N terminus to C terminus. In a protein there are 20 possible amino acids, but in a DNA molecule there are only 4 possible nucleotides, whose one-letter codes are A, T, G, and C. The full names of these nucleotides are adenylate, thymidylate, guanylate, and citidylate. Now, it turns out that in the cell we don't have one DNA strand on its own, but it exists bound to a second strand like this. Now, there are a few things to notice here. One is that the strands run 5' to 3' in the opposite direction. This is called antiparallel. The other is that A always pairs with T and G always pairs with C. This is called base pairing. So if you have the sequence of one DNA strand, you should be able to write the sequence and direction of the other. Now, we know that to get to protein we have to carry out transcription, where the information from DNA is copied to RNA. The RNA molecule we just made is called messenger RNA, or mRNA for short. The sequence of the mRNA is what you would expect if it base paired with the bottom DNA strand here, except that where we would have T or thymidylate in DNA, here we have U or uridylate. In fact, the mRNA molecule is made through base pairing to the bottom DNA strand, and this strand is called the template strand. The reason it is called that is because it is used as a template in the sense that RNA nucleotides physically interact with the template strand as it is being made. The other DNA strand is called the coding strand. It is called that because while it is not used as a template, its sequence is the same as the resulting mRNA, except that it has thymidylate instead of uridylate. The name of these strands can be a bit confusing, but when we go over how nucleic acids are made, it will make a bit more sense. As we saw before, from RNA, the information is used to make protein in a process called translation. But how do the nucleotides in the mRNA specify a protein sequence? Well, it turns out that groups of 3 nucleotides, called codons, specify a certain amino acid. The scheme of which codons specify which amino acids is called the genetic code. We can also see here that reading the mRNA from the 5' end to the 3' end represents the protein sequence going from the N terminus to the C terminus. Okay, so at this stage we know that nucleic acids like DNA and RNA are made of nucleotides. But what is a nucleotide? Well, there are 3 basic parts. We have phosphate. This is the same in all nucleotides. Then there is a pentose sugar attached to the phosphate. If we look at how we number the carbon atoms in the sugar, you can see why the ends of nucleic acid strands are called 5' and 3'. It is because nucleotides are joined together through these positions, and therefore one end of the chain will have a 5' phosphate and the other a 3' hydroxyl, or OH. Now, the sugar I'm showing here is ribose, and this is the sugar found in RNA. And so this means that DNA and RNA have nucleotides with different sugars in their nucleotides. But then what sugar do DNA nucleotides have? Well, they have deoxyribose instead of ribose, which is why the full name of DNA is deoxyribonucleic acid. Now these look pretty similar, but they have one difference that you should be aware of, which is that ribose has an OH group on the 2' position, whereas DNA doesn't. The final part of nucleotides is called a base. I'm just showing this as a box right now. It is joined to the sugar's 1' position, and the bond is going up from the sugar as it is drawn here. Because this bond is pointing up from the sugar like this, it is called a beta glycosidic bond. You will learn a lot more about this during the carbohydrate section of the course, but for now I will just say that if the bond was pointing down, it would be called an alpha glycosidic bond. The type of base controls what one-letter abbreviation we use when we write out nucleotide sequences, and in DNA, the choices are A, G, C, and T. In RNA, the choices are A, G, C, and U. So we know there are a bunch of bases that can be incorporated into nucleotides, but why is it important to understand their structural differences? Well, first, the base order in nucleic acids encodes information, either protein sequences or other things. And the structure of each base controls which other base it can pair with. And as we shall see later, this is the mechanism which allows nucleic acid strands to be copied. Here I'm showing the structure of all 5 bases used in DNA and RNA. Their names sound a bit like the nucleotide names, but slightly different. We have adenine, guanine, cytosine, uracil, and thymine, with one-letter abbreviations as you would expect. Now, one thing you should be aware of is that these bases fall into 2 groups. The ones with 2 heterocyclic rings are called purines, and the ones with 1 heterocyclic ring are called pyrimidines. Now, it will be useful in this course for you to know which bases are purines versus pyrimidines, and be able to tell the difference between a purine and a pyrimidine structure. So here are some useful mnemonics. The first is the phrase pure as gold, which you can use to remember that adenine and guanine are purines. The second is the phrase pyramids are cut from pure stone, which may help you remember that pyrimidines are smaller than purines and therefore have 1 ring, with purines having 2, and also that cytosine, uracil, and thymine are pyrimidines. These methods may or may not work for you, but if you want ideas on how to remember biochemistry concepts, you can often find other mnemonics that people found useful on Google. So once again, these are the bases, and I'm showing their names as well. If we add a ribose onto these bases, then we now have ribonucleosides, or just nucleosides. Notice here that this name has an S. We are talking about nucleosides here, where there is just a base and a sugar, whereas nucleotides also have the phosphate. The name of each ribonucleoside is also different to the base, as they now end in osine or idine. If we add a phosphate onto the 5' position of these ribonucleosides, then we have ribonucleotides. Again, they have different names now, ending in -ate. And additionally, they have 3-letter abbreviations ending in NP. For example, adenylate is abbreviated to adenosine monophosphate, so the nucleoside name plus monophosphate. An interesting thing to note here is that ATP, the molecule that is used to store chemical energy in the cell, is adenosine triphosphate, so basically the same as AMP, but with 3 phosphates strung together on the 5' position instead of just 1. And this tells us that in addition to storing information in nucleic acids, nucleotides can also be used to store chemical energy to drive reactions, as we will see in this section and also in the carbohydrate section in the course. But one thing you may have noticed is that we have just seen the ribose forms of these compounds used for RNA. So let's go back to the bases, but in this instance we have thymine instead of uracil because we are thinking about DNA rather than RNA now. So when we add deoxyribose now instead of ribose, these become deoxyribonucleosides, with deoxy indicating that we have deoxyribose, and the S instead of T, because we just have a base and a sugar with no phosphate. The individual names are deoxy variants of the ribonucleoside variants we saw before, with one quirk, which is that instead of deoxythymidine, the deoxyribonucleoside of thymine is just called thymidine. This is because thymine is only found in DNA, and so we would never see the ribo form of thymidine. For bases used in RNA, if you don't see deoxy in the nucleoside or nucleotide name, you can assume that ribose is being used, but for thymine, the default form uses deoxyribose. When we add a phosphate to the 5' position of these, we then get deoxynucleotides. And this is entirely in line with what we have seen before. The names now end in -ate, for example deoxyadenylate, and are abbreviated as before, for example deoxyadenosine monophosphate. Let's now think about how nucleotides are joined together. Here we have the structure of a deoxyribonucleotide, and as I said before, when this is in a DNA strand, it is joined to other deoxyribonucleotides through the 5' and 3' positions. As you can see, one end of the strand has a 5' phosphate and the other has a 3' OH, and that is why we call one end of the strand 5' and the other 3'. You can also see that the nucleotides are joined through phosphates. We call this structure of a phosphate with substituents on two of the oxygens a phosphodiester. That is why we call this continuous stretch of bonds with many phosphodiesters the phosphodiester backbone. Why is this important? Well, phosphodiesters have a pKa of about 2, and this means that at the physiological pH of 7.4, they are negatively charged. This strong charge increases the solubility of DNA, which helps proteins interact with it. The negative charge also repels nucleophiles, which are often also negatively charged. Another way of saying this is that the phosphodiester backbone is stable to hydrolysis, meaning breakdown by water. In water there is also a certain amount of OH- ions, and they can attack things with some of their negatively charged electrons. But when the backbone is strongly negatively charged, this will be prevented because negative charges repel other negative charges. This stability is important because we want our genetic information to stay safe, and we don't want it easily broken down. As well as being charged, DNA molecules are generally large, and these two properties together prevent them from diffusing through membranes. This is important because we want our genetic material to stay in the cell and not float away. So we already learned that DNA is stable to hydrolysis, but we also know that RNA is not very stable at all. Why is that? Well, the answer lies in the chemical structure of RNA. RNA, of course, has nucleotides that contain ribose, which has this extra OH on the 2' position that is not seen in DNA. This is the key to understanding the difference in stability in RNA versus DNA. But before we go into that, I just need to go through a few basic things about chemical mechanisms. Now, I know, of course, that organic chemistry is a big subject, and it can also be intimidating. But I'll just go through the bare minimum that would be helpful to understand the material in this course. In chemistry and biochemistry, we are concerned with how atoms interact to make new covalent bonds. You can think of atoms as being made of two parts, a central nucleus and shells made of electrons. The nucleus consists of protons, which are positively charged, and neutrons, which are neutral. Consequently, nuclei are positively charged. Electrons are negatively charged. Here I'm showing a very simple atom, deuterium, which is basically just hydrogen but with an extra neutron in its nucleus. A covalent bond is drawn as a line like this, and you can think of it as two atoms sharing a pair of electrons. Sometimes you get a molecule that is negatively charged or has enough spare electrons that it likes to make new covalent bonds with other atoms. We call this type of molecule a nucleophile because it loves to push its negatively charged electrons towards the nuclei of other atoms, which are positively charged. Literally, this means nucleus loving. You can think of this as an electrostatic interaction of positive and negative charges. There are also other molecules such as acetone here that like to accept electrons, and therefore we call them electrophiles, literally electron loving. Now in chemistry we represent how a reaction happens, its mechanism, with curly arrows, which indicate the movement of pairs of electrons. So here we show the hydroxide pushing electrons to the central carbon atom of the acetone. This forms a new covalent bond. But of course, those of you who know anything about chemistry can see that this structure is all wrong. The central carbon has 5 bonds going from it, as there are 3 single bonds and 1 double bond. That of course can't happen because carbon can only have a maximum of 4 bonds. So in this reaction, 2 electrons from the double bond migrate to the oxygen. Then they manifest as an extra negative charge around the oxygen. Now it is easy to imagine that things with a positive charge attract electrons and are therefore electrophiles, but in this case acetone is an electrophile because it is able to push some electrons to the oxygen in this way. So in water you get a certain amount of hydroxide ions floating around, and they can act as nucleophiles in hydrolysis reactions, which means the breaking down of molecules with water. However, with our RNA structure, we know that hydroxide cannot attack the phosphate directly because the phosphate is far too negatively charged to be an electrophile. However, it turns out that most of the negative charge is concentrated around these phosphate oxygens, while the oxygen atom of the 2' OH is positioned really close to the phosphorus atom. Hydroxide cannot directly attack the phosphorus, but it can attack the hydrogen on the 2' OH to form a bond. Hydrogen can only bond to one other atom, so the electrons in this existing bond have to go somewhere. And because the 2' oxygen is rigidly held close to the phosphorus atom, it can form a bond with it. Now, just like carbon can't have more than 4 bonds going to it, phosphorus can't have more than 5 bonds going to it. And in this unrealistic structure here, it has 6. So one of the bonds has to break, and in the process we break the phosphodiester backbone, and we make this 2', 3' cyclic derivative. And so this is why RNA is much more hydrolytically unstable than DNA. So at the end of the video, what have we learned? After going through this material, you should be able to know the pathways of information flow originally proposed in the Central Dogma. You should be able to say whether a base structure is a purine or pyrimidine and know which bases fall into which category. You should be able to identify a given structure as either a base, nucleoside or nucleotide and know the difference between the ribo and deoxyribo variants. You should know which bases pair together in double-stranded nucleic acids. And you should be able to look at the structure of a DNA or RNA molecule and point out the 5' and 3' ends. And you should also be able to explain how RNA is much more unstable than DNA. And if you want to read more about this, I've posted PDFs of Leninger chapters 8 and 24 on Canvas.